Hello everyone, and welcome to our lecture on Chapter 2 in Physiological Psychology. Here we're going to talk about nerve cells, the structure and function of them. We'll talk about neurons first, then we'll talk about the support cells, which are generally called glia. Then we'll talk about communication within and between neurons. The signals that are generated inside of neurons are going to be electrical in nature, and the signals that get passed between neurons are going to be chemical in nature. What you can see in this cartoon here on our first slide, we have our yellow neuron surrounded by a few different support cells. The astrocytes are red, oligodendrocytes are blue. They perform different functions, and the chemical signals are being transmitted between two of these neurons on the right there. So we'll cover a little bit more detail, but this is the general idea of what we're talking about today. We'll start with neurons, since they're kind of the star of the show here. Neurons are polar cells that are specialized to be highly excitable and rapidly communicate signals within themselves and then between them and their target cell, which it could be another neuron, it could be a muscle cell, for example, it could be a gland. So neurons will talk with other cells besides other neurons. Okay, when we say that neurons are polar, we mean that they have a sidedness, just like a magnet has a plus and a minus end. Neurons have a receiving and a shipping end. In the center of the cell, we have the cell body, which is also called the soma. And this is where a lot of housekeeping tasks are carried out. Proteins are created. The signals that are received in the dendrites are going to get integrated or added together to determine whether or not another signal should be sent down the axon. As I mentioned, the dendrites are receiving those signals. Okay, so the dendrites are these little tapered extensions that are typically responsible for receiving the synaptic input for a neuron, although synapses, as we'll see, can form on axons and they can form on cell bodies as well. But we tend to think of them on dendrites because dendrites have such a huge surface area. So the dendrites receive signals, those signals funnel into the cell body, and if they are sufficiently positive, then it will cause the neuron to fire an action potential down its axon. The axon is going to be a uniform diameter, there's no tapering, but it will branch extensively in some cases in order to innervate multiple targets. As you can see in the little cartoon below, that yellow neuron is receiving input from a good number of blue neurons there, roughly 4 by my count, but who knows. Those signals are getting integrated at the cell body. It was sufficient to excite it, and then it sent a signal down its axon. You can see it branches once in this cartoon. Of course, in real life, it branches many, many more times, and so this way it can communicate with 2 other neurons. Now, those sites that are linking our nerve cells are called synapses, and what they look like at the microscopic level would be something like this. On our left there, we have our presynaptic terminal, and you can tell that because of all those little tiny circles in there. Those are vesicles filled with the chemical signals that will be passed between our presynaptic site on the left and that postsynaptic site labeled accordingly in the center of this illustration. Now, surrounding this synapse, you can see the yellow pseudo-colored process of an astrocyte. That's a glial cell. It's surrounding the synapse, and as we'll find out soon, what that astrocyte is doing is removing the neurotransmitter if it leaks out of the synapse so that it can maintain a very low level of noise at the synapse. Very important function, but we'll touch on that in a bit. You can always tell which is the presynaptic terminal, though, by the presence of those vesicles, because as we'll see, neurotransmitters are always stored in vesicles. The cell body is very much your prototypical cell. It has the nucleus where transcription will take place. It has all the endomembrane system, so it has the ER and the Golgi, so it can carry out protein synthesis and package it and distribute it accordingly within the cell. The cell body is pretty much all about protein synthesis. That's kind of its major function. Electrical signals will diffuse through it passively, but there's not a whole lot of active processing going on that we need to talk about yet. You'll also find some protein synthesis in the dendrites. There is local protein synthesis. It's likely very important for our ability to store information. I don't know if we'll talk about the details of that in this class, but keep in mind protein synthesis can also take place to some degree in the dendrites, but it's a lot more restricted. It's only very specific proteins that are being created there. You won't find any protein synthesis in the axons, though. Axons are completely devoid. We can tell that by simply doing a Nissl stain. That's what we see on the bottom here. So everything that's colored purple is showing you essentially the presence of ribosomes. That's why the nucleolus is so dense there, since that's where we create our ribosomes. So the purple Nissl substance is very dense in the cell body. You can see that the dendrites are still faintly purple. That's because they are capable of carrying out protein synthesis. And once you hit the axon hillock, that's where the axon connects to the cell body. You can see that in the bottom left of that illustration. It's the same as background signal. So there's really no ribosomes being carried down the axon hillock. So no protein synthesis. Now the number of dendrites that a neuron has is going to be unique to that neuron. Every neuron is going to be a little different. Some will have 4 dendrites coming off the cell body. Some will have dozens. Okay, and those dendrites that come off the cell body are what we call primary dendrites. Once they branch, they create secondary dendrites. When those branch, they create tertiary dendrites. But one of the ways that we can characterize neurons is by the number of primary dendrites. Whereas a pyramidal neuron will have somewhere in the ballpark of 4 or 6 primary dendrites. Your stellate neurons, which tend to be local interneurons, those are going to have numerous dendrites located uniformly around the cell body. So on that left illustration, you can see that PY, that pyramidal neuron, has a very different look to it than the interneuron next to it, that INT. One is pyramidal, one is stellate. Stellate just means it looks like a star. Not every dendrite will branch the same. Some will branch many times. Some will branch relatively few times. Some will branch near the cell body. Some will branch very far from the cell body. So this is yet another way of characterizing the dendrites. The last thing that we use to, the last thing on this slide that we use to characterize dendrites would be the presence or absence of dendritic spines. And those are little tiny thorn-like projections that come off the dendrites. These increase the surface area of the dendrites immensely, and they also create an area with incredible internal resistance, which can amplify those electrical signals, which we really won't talk about in much detail in this class. But dendritic spines are very important. They can be transient structures. They can grow and shrink, allowing us to selectively strengthen specific connections and thus learn or unlearn information. Dendritic spines are very, very important. We won't give them the attention that they deserve in this class, though, unfortunately. Okay, moving on to the axon. Here's where we transmit signals. Okay, so remember, we're going to receive signals, for the most part, in our dendrites. Those electrical signals generated in the dendrites will funnel into the cell body or the soma, and if the neuron has been excited sufficiently, it will transmit another signal down its axon. Now the axon is joined to the cell body at a region that we call the axon hillock. This is loaded with sodium channels, which, as we will see, are absolutely fundamental for generating positive electrical current in our neurons. The axon itself is actually fairly small in diameter, despite what this hideous cartoon is showing you. It's small in diameter, and it can traverse very long distances or very short distances. The neuron on the left there, we're looking at the dendrites colored in blue, and the axon is in red. You can see that the axon is actually covering a much larger area than the dendrites, despite what cartoons will often suggest of neurons. The axon is usually much, much bigger than the dendrites. Even on a local circuit neuron, which doesn't project far away, that'd be the neuron on the right. In this case, the dendrites are colored red and the axon is colored black. So even though the axon is not really going that far from the cell body, it's still covering a much larger area than the dendrites. These axons may or may not be myelinated. If they are, then they will conduct their signals much more rapidly than an unmyelinated axon. We'll talk about myelin once we discuss oligodendrocytes in Schwann cells, because the myelin is not actually created by neurons. Instead, it's created by one of those support cells. If you're in the central nervous system, it'll be an oligodendrocyte, and if you're outside, it'll be the Schwann cell. But that myelin is just a fatty substance that insulates the axon so that it can send its signals more rapidly. And those signals would be the action potential. That's what we're looking at here. We'll talk about this a bit more in this lecture. So the neuron, just like every other cell in your body, has a charge across its membrane. That's what we call the membrane potential. Your typical resting neuron, we're going to say, rests at negative 70 millivolts. That means that it has a negative charge relative to the fluid surrounding it. Part of this has to do with the DNA and phosphate-containing proteins, because phosphate can't leave the cell and it has a negative charge that kind of biases the cell toward this negative membrane potential. But we'll see that there are many ions that actually contribute to the cell's membrane potential, and that it's not a fixed value. The membrane potential is not always negative 70 millivolts. In fact, in some cells they rest at a different value. The one in this cartoon is resting at negative 62 millivolts. That's okay. But it's negative. Okay, it's always negative for your neurons and their support cells, and pretty much every cell in our body. During an action potential, though, the membrane potential will briefly become positive. Okay, so remember, in the cell body, we're summing those signals from our dendrites. Maybe we receive just a little bit of excitatory input in our dendrites. That's what's going on in the red line there. You can see that there's a little bit of an upstep. It becomes a little bit more positive than negative 62 millivolts, but it doesn't do much after that. It goes back down to rest fairly quickly. If we receive sufficient excitation, that's what's going on in that purple current there that's actually creating the action potential. So once we reach our threshold potential, which we will talk about in a bit, that's going to open up those sodium channels that litter the axon and the axon hillock. Sodium is going to rush in. We're going to actually achieve a positive value in our membrane potential, and that's going to be very important for stimulating the release of chemical signals between neurons. The action potential has built-in mechanisms for returning the membrane potential back down to the resting negative value. In fact, it goes a little bit more negative than it was originally, and then it will return back to its minus 70 in a textbook neuron. Action potentials are those electrical signals generated within cells. The neurotransmitters are the signals sent between cells in response to the action potential. Okay, we're going to talk more on the action potential in the third section of this lecture. Now, in order for a neuron to have its exquisite shape, it needs to have some sort of structure, and that structure would be the cytoskeleton. And really there are three parts of the cytoskeleton. There's the microtubules, there's the microfilaments, which are made of actin, and then there are the intermediate filaments. We're not going to talk a whole lot about here. I assume that you've had a cell biology class, so we won't review this in any great detail. But the microtubules themselves are polar structures. They are oriented such that they're negative ends. Again, polar just means it has a sidedness. The negative ends are located usually toward the center of the cell, except when they're not, as we'll see. But in general, negative ends are attached to the centrosome. That's our microtubule organizing center, and they radiate out. In these cells that we're looking at here, we can see our actin cytoskeleton is purple, the nucleus is blue, and those yellow strands, those yellow ropes, would be the microtubules. And you can see they're all kind of radiating out from the center there. That's where the centrosome is. The positive end of the microtubule thus always points toward the end of the cell, except with dendrites. These are a notable exception to that microtubule directionality. In the dendrites, the positive end could be pointing away from the cell body. It could be pointing toward it. So this kind of leaves some open questions about how do cells and their proteins know which direction they're moving in the dendrites. How do they actually know how to move something away from the cell body when their microtubules are all oriented in different directions? And this is this is still an open question. We do understand a good deal about axon transport because the microtubules are very orderly. The minus end is toward the cell body, the plus end will be toward the end of the axon and thus toward the synapse. So when a neuron goes through the trouble of making proteins that are destined for the synapse, it's a very simple job. Attach them to a motor protein. If you want to move toward the synapse, attach them to kinesin. That's the motor protein that moves toward the plus end. If you want to take them back to the cell body, attach them to dynein. Dynein is the motor protein that's always going to run toward the minus end and thus toward the cell body. Those two motor proteins are going to burn through ATP in order to walk in one direction along the microtubule. Hopefully it's going to record this video. I've had some issues with recording it, but if not, just follow the link on the bottom and what you can see are these little vesicles dancing along this cellular process. So on the top we're looking at dilated vesicles. Those little balls are little spheres of membrane filled with a dye. And you can see some are moving to the right, some are moving to the left. The ones that are moving to the right are on kinesin, the ones that are moving to the left are on dynein. So some vesicles are moving away from the cell body, and others are moving toward it. That central view, that's just light microscopy. You can actually see cell movement in it, but of course it looks best on the bottom. That's where you can see the actual cellular process overlaid with the vesicles moving by. And so those little spheres are attached to little tiny proteins that are just moving along the microtubules by burning ATP. So this is how a neuron with its cell body in our spinal cord can still send proteins down to a synapse, maybe in our big toe, to allow us to actually control muscles, even though they're very, very far away from the cell body. It takes some time, but because of the directionality of microtubules, the cargo will always get to where it needs to go. Okay, let's talk glia. Glia just means glue. Okay, so glial cells they're the cells that kind of hold our brain together. That's what we thought anyway. This is an oversimplification. They're going to support the function of neurons in a variety of ways. Our oligodendrocytes and Schwann cells, they create myelin. That's that fatty substance. Astrocytes help maintain homeostasis by buffering neurotransmitters and ions and supplying metabolic support to neurons. And microglia are going to act as the immune system of the brain. We'll start off with our two myelinating cells. We have the oligodendrocytes and the Schwann cells. In the central nervous system, so in our brain and spinal cord, oligodendrocytes are responsible for generating myelin. In the peripheral nervous system, so with our motor nerves and things like that, the part of our nervous system that actually reaches out to our muscle, those are controlled. Those are myelinated by Schwann cells. Okay, this illustration is showing us the cross-section of an axon. That's what AX means. So we're looking at the center of an axon, and you can see that it's surrounded by these concentric layers labeled MY. That's the myelin. So these are just layers upon layers of fat. The MIT in there is a mitochondrion. That's going to obviously generate ATP to help keep the neuron fueled. So myelin is going to wrap around the axon to help increase the rate at which we can communicate. Because in order for our nervous system to function properly, we need to be able to send signals rapidly. And there's two ways of accomplishing that. You can lower the internal resistance by increasing the diameter of your axon, or you can increase the membrane resistance by ensheathing it with this fatty substance. So myelination actually helps us pack more neurons into a smaller area and thus have more complicated brains than our invertebrate counterparts. What myelination does is cause the action potential to kind of jump along the axon in what we call saltatory conduction. So there are two different regions now if we have myelination. It's not just a naked axon. There are exposed regions of the axon that we call nodes of Ranvier, but then there are the myelinated regions. Those would be the internodes because they are between the nodes of Ranvier. So the internodes have incredibly high membrane resistance. Essentially no ions will cross the membrane. So any electricity that we generated in that action potential is going to flow passively and very rapidly down the axon to the next node of Ranvier. At each node of Ranvier, we have ion channels. so we can generate more electrical activity, because that passive diffusion actually dissipates the amount of charge to some degree. And if we're going to send a signal from our spinal cord down to our big toe, we need to regenerate this electrical signal so it doesn't diminish to nothing by the time it gets there. So our internodes speed up the conduction, and then our nodes of Ranvier amplify the signal back to its maximum value. So they replenish that electrical signal so that it can diffuse passively in the next internode. And that's the skipping along. So it sort of skips quickly through the internode, pauses briefly at the node of Ranvier to recharge, and then skips along. While oligodendrocytes and Schwann cells both accomplish the same task of generating myelin, they do so in different ways. The oligodendrocyte creates myelin in a manner that conserves space. It's going to myelinate multiple segments of multiple axons, and it's going to create a very dense form of myelin, highly compacted, not a lot of cushioning going on here, because it's in the central nervous system. That's going to be protected by our skull and some layers of fluid, the spinal cord protected by our vertebra. So we don't need to provide as much cushioning here. What we need to do is figure out how to pack as many neurons and other cells as possible into a head that doesn't occupy the size of the room. So we want to have small heads so we're mobile, but we still want to pack neurons in there. That's going to require a very compacted myelin with not a lot of cushioning. The peripheral nervous system, on the other hand, has space. We can have our nerves swell up a little bit, and in fact it's a good thing that we have that swollen, cushiony myelin that Schwann cells generate, because nerves in the peripheral nervous system aren't protected by a skull or vertebra. They're exposed to the world, and as you're bumping through life, especially early on, there's a lot of bumping involved. You're hitting your nerves. And if you're to damage the nerve and sever the axon, then you're going to experience at least temporary paralysis, if not permanent paralysis. So the Schwann cell creates sort of a fluffier, more cushiony form of myelin that's going to protect the axon, as well as increase the rate of action potential conduction. So oligodendrocytes will myelinate multiple axon segments, but the Schwann cell is only going to wrap around a single segment of a single axon. So you can see in the top, the oligodendrocyte there is surrounding just those three little segments, but the Schwann cell is going to just surround the one. If we were to unroll a Schwann cell, we'd see that it just kind of wraps around one little section of axon. So the Schwann cell is the kind of trapezoidal cell there, and the axons running along the bottom. Schwann cells are also going to be capable of removing cell debris. So if you do have some cellular damage, they can clean it up and release growth factors to help us regrow our damaged axons to some degree. Okay, we can regenerate peripheral nerves somewhat. Damage in the central nervous system is much more drastic because oligodendrocytes do not help clean up or promote the growth of axons. So a spinal cord injury, permanent. If you were to damage a nerve in your arm, you may lose some degree of motor control and feeling, but some of it will come back. It could come back completely. You might be left with a slight tingly feeling whenever you touch your hand or something like that, but you can still have some regeneration. And that's because of differences in those two glial cells. Moving on to really the star of the glial cells, that'd be the astrocyte, which means star cell. These do a number of really important functions to help our central nervous system maintain homeostasis. So you're going to find these in our brain and our spinal cord. They're present as we develop. They're going to help the neurons find their way by secreting guidance molecules and growth factors to help neurons adopt the correct morphology and to differentiate into the correct type of cell, as well as survive. They're also very important for clearing neurotransmitters, namely glutamate and GABA. These are really the most common neurotransmitters in our brain. They're ubiquitous. You find them everywhere in the brain. Glutamate is excitatory, GABA is inhibitory, and astrocytes have the appropriate transporter proteins to pick them up. So here's our synapse that we looked at before. Now it's in complete cartoon format. The presynaptic site is purple on the yellow. It's got its little yellow vesicles filled with glutamate, it looks like in this case. The postsynaptic site is still in the center. Now it's kind of a light blue there. So when those vesicles fuse, if we have an action potential, they will. They'll spit out the neurotransmitter and it will bind to receptors on the postsynaptic site. But what happens after that? Is it just going to hang around forever and continually excite that neuron? I hope not. Okay, because bad things happen. That can actually kill the neuron if it's overexcited. It's what we call excitotoxicity. And if it doesn't do that, then it just makes it difficult to hear the next signal. If we don't clean up that glutamate, then we're going to have this constant activation of glutamate receptors. If we dump more glutamate out, it's hard for the postsynaptic cell to tease out that there's been a new signal. So instead what we do is rapidly remove that glutamate from the synapse. Neurons have glutamate transporters. They're largely ineffective compared to astrocytic transporters. The transporters on astrocytes are actually going to kind of wait for the glutamate to diffuse out of the synapse, and as it's leaving, because astrocytes are located at the periphery there, it's going to remove that glutamate. It'll also buffer some ions, but the glutamate buffering is really critical because without this, we get protracted and lethal seizures. At least mice do. Some of these unlucky mice were born without glutamate transporters in their astrocytes. This was by design, so they're glutamate transporter knockout mice. And since we have different transporters in neurons and astrocytes, they could distinguish between those two transporters. So they knocked out the astrocytic transporters, and in panel A, what we're looking at is just body weight, just the average body weight, so the growth of the mice. And you can see it works fairly well until about week between 20 and 30, they start to differentiate. So the open circles, those would be the wild type mice that have their glutamate transporter. You can see that those climb up to a nice healthy 25 to 28 grams, whereas the black circles there, those are the knockout mice. You can see they don't generate as much body weight. They don't really reach 20 grams on average. And while that's not a good thing, it gets much worse in panel B. Panel B is showing the survival. In a wild-type mouse, same coloration here, the open circles are staying around 100%, and mice should certainly live well beyond 12 weeks. The black circles, it's a very different story. So the knockout mice, they have very rapid death. You can see that they start to die off after about 2 weeks, and by the time we hit 6 weeks or so, about half of them are dead. That's not normal. The reason that they die is because of these protracted seizures that you can see in panel C. That's a little mouse having a seizure, and really all the seizure is widespread hyperactivity in nerve cells. And we can measure that with EEG by attaching little electrodes onto the scalp. We can measure activity in the brain. The top trace there is showing you very small fluctuations, which you would expect in a normal mouse, and that's what they're recording from. The bottom trace there, you can see much larger up and downward deflections. So those bigger squiggles are showing you these epileptiform or seizure-like periods of activity. So you see a lot more hyperexcitability of nerve cells, which lead to seizures and death. So without our astrocytes functioning properly, we don't function properly. All right, and that's what makes them the star, not just their name. Now, astrocytes, just like neurons, have very complex morphologies. They don't just wrap around axons. They will wrap around axons. They'll also wrap around cell bodies and dendrites. Typically, what people look for is one of their cytoskeletal components called GFAP, glial fibrillary acidic protein, that's shown in red. And when you look at this, you see, okay, they're occupying very discrete areas. Sort of, not really. If you fill the cell up with biocytin, that's just this little diffusible molecule that you can then label with an antibody. That's shown in green. You can see that it's occupying essentially the entirety of this field of view except for that little neuronal cell body on the left there. Now, these astrocytes don't just operate in isolation. In fact, they form very large networks containing numerous astrocytes, so that whenever one astrocyte picks up a neurotransmitter, it can then send that throughout the entire astrocytic network to increase the buffering capacity. So rather than just having one sponge to soak up the glutamate, you now have hundreds of sponges, which means you can soak up a whole lot more glutamate. Astrocytes are going to surround axons, dendrites, and cell bodies. Like I said, they also surround blood vessels. Okay, now we're looking at GFAP, but it's not red. It's white in this top illustration, so that's showing us where astrocytes are. And you can see that they do have some extensions that are just kind of radiating out, but there's clearly an organized structure here. There's this blood vessel running from the bottom left corner up through the middle of the field of view here. And all the astrocytes are kind of surrounding that blood vessel. They're helping form what we call a blood-brain barrier, so they surround essentially the entirety of the blood vessels in our brain and make sure that anything that's inside the blood that might be toxic stays in there, because neurons are very delicate. The other extensions are going to surround neuronal processes so that the astrocyte can buffer neurotransmitters, like I said, and ions released at the nodes of Ranvier, but also so that the astrocyte can actually send molecules from the blood into neurons. Now, removal of molecules from the blood must be very carefully controlled, because sometimes we put toxic things into our blood because toxic things can be fun. Also, sometimes we just make toxic things as a part of our normal metabolism. So to prevent our delicate nerve cells from dying every time we want to have a party, we have this blood-brain barrier that kind of keeps toxic components out of the blood. And the blood-brain barrier is just a collection of tight junctions that restrict the flow of molecules out of our blood and into the brain. So only very small molecules or very hydrophobic molecules are going to freely pass the blood-brain barrier. The rest of them are going to have to move through in a controlled manner. The blood-brain barrier is very important. It does get damaged in some cases of severe head injury or neurodegenerative disease. And once that happens, then you have another host of problems because of all the blood-borne toxins. So part of the issue with head injuries lies in the disruption of the blood-brain barrier. This thing is very important because under normal circumstances, when it's working well, it restricts the flow of molecules. So now you have very controlled movement. There are some molecules that freely flow across. Those would be things like gases. So our oxygen and carbon dioxide, they have no issue crossing the blood-brain barrier. They freely flow through membranes. Other molecules are going to need some sort of transporter, some sort of protein to help guide them through. All right, and you're not going to have to know all of these different mechanisms. We should understand, though, that there are transport proteins for very critical molecules, like glutamate, for example. Glucose, sorry. Glucose is going to be moved across the blood-brain barrier through glucose transporters, which would be the glutes. So if we're going to get the sugar from our blood into our brain, we're going to have to actually move that glucose because it can't move freely. Right? Our blood-brain barrier is restricting its movement. So we have to have glucose transport proteins. Of course, we're going to need them on the epithelial cells. Those are those blue cells in this cartoon. So that would be your blood vessel. There's the edge of the blood vessel. Blood's on top. Our brain is on the bottom. And those astrocytic end foot processes, the parts of the astrocyte that surround the blood vessels, they also need glucose transporters, because ultimately glucose is what fuels our brain. So without glucose, our brain will run out of energy. That means that we have to have those glucose transporters in our vascular epithelium and on our astrocytes and then on neurons. So the astrocytes that surround the blood vessel, we can see that in the top panel there. They're going to remove glucose. Those astrocytes are also near neurons. So if we look in that bottom panel, we can see our little green astrocytes. They're little clouds. Remember when you fill them with biocytin, they kind of fill up the whole area. There's 1, 2, 3, 4, 5 of them surrounding this red neuron here. And there are more of them, they're just not labeled. But those astrocytes, they take glucose from the blood supply and then they shuttle it to neurons. Sometimes they'll give the glucose as is, other times they'll convert it to lactate after undergoing anaerobic metabolism and then send that lactate to the neurons where they can very rapidly use that in aerobic metabolism. So astrocytes are going to help make and maintain that blood-brain barrier, and they're going to help shuttle those metabolic substrates like glucose and lactate from the blood to the neuron. All right, so astrocytes are very important for fueling neurons in addition to cleaning up the mess that they make as they talk to one another. The last glial cell that we need to talk about would be the microglia. They are the macrophages of the brain. We're looking at microglia under two different conditions here in this illustration. On the left, we have normal microglia. Nothing has happened to this brain. They are resting. You can see that they have little cell bodies and they have little tiny extensions coming off of them. They look kind of like little stellate neurons, except they're much smaller. On the right, this brain has been injured. We can see a couple of things going on here. One, the microglia have a very different morphology. They're kind of a more amoeboid here. They look like a little amoeba. So they're rounded up. That allows them to migrate. And that lets us see the second thing going on here. There are a lot more microglia in this area. So microglia will travel to sites of injury and help remove cell debris. And in order to do that, they need to retract their cell processes and then wiggle their way into the site of injury. Once they clean up the injury, they're going to secrete some growth factors and cytokines to help synapses regenerate, new glial cells to form, and in some cases make blood vessels as well. But whenever we think microglia, we should be thinking macrophage because we don't have an immune system in our brain. We have the blood-brain barrier. That's going to restrict the movement of molecules, including the antibodies and the immune cells that are responsible for keeping our periphery clean. So if we were to get an infection in the brain, it would be screwed without the microglia. But we have this resident immune system in our brain, and that's the microglia. So oligodendrocytes and Schwann cells create myelin. Astrocytes help remove neurotransmitters and supply metabolic substrates to neurons, and then the microglia are going to act as the immune system. So with all of our glia and neurons together, now we have a properly functioning nervous system. So now we can talk about the signaling that takes place within and between neurons. Within a neuron, it's all about generating electrical signals. And we can do this because all neurons and all cells in our body have a charge across their membrane. It's called the membrane potential. You've learned about potentials in your physics classes. This is the exact same thing. It's a separation of charge. That's all it is, and it's a separation of charge across the membrane. Now, when a neuron's not doing anything, we say it's at rest, and we assume that it's somewhere near negative 70 millivolts. It could be negative 80. It could be, negative 50. Totally depends on the neuron. Now, despite having this difference in charge, we have to have equal osmolarity on both sides. Otherwise, the cell's going to swell up or shrivel, depending on if we have too many ions inside or too few ions inside. Okay. Now, even though we have the same number of ions inside and outside, what we don't have is the exact same amount of each ion. Inside the cell, if we start at the left here, you can see that A minus, that's the impermeable anions. Those would be those phosphates that make up our DNA and get attached to proteins. They're also present on ATP. Those phosphates have negative charge and they do not leave the cell. So those remain inside and they bias the cell toward a negative membrane potential. Potassium, that'd be the K plus there, that's a cation. It has a positive charge, and you find a whole lot more of it inside the cell than outside. On the other hand, sodium and chloride, in other words salt, it's what you'd find in salt water, that's a lot higher outside of cells, and this probably has to do with the origin of cells being born in that saltwater environment. So there's a lot more sodium chloride outside the cell, and there's a lot more potassium inside. There are many other ions that we're not talking about, but this is sufficient. If you know potassium, sodium, and chloride, you're good to go. Okay, now how do we measure the charge in a membrane? Well, we do that by using electrodes. You can actually pierce some cells with an electrode and they'll live, and you can record electrical activity. For other cells, you have to be a little more delicate. You have to use patch clamp recordings. And in a patch clamp recording, you have a little tiny glass pipette that's been polished so it's nice and smooth. It won't poke the membrane. You can actually attach it to the cell membrane. That's what we see in the top left there, cell attached. You don't open up a hole in the membrane. These are very noisy recordings, and you can only see big changes, like action potentials. But it doesn't give you a very accurate reading of what's going on inside the cell. What you have to do is apply a little bit of negative pressure, so you have to give a little suction. That'll pull open the membrane, and now the inside of your pipette is continuous with the inside of the cell, and you have exquisite control over the membrane potential if you want to control it, or you can just simply record it. So when you go whole cell, there's a couple things going on. So your electrode is going to be attached to some very simple circuitry that's going to measure the voltage across the membrane relative to some reference electrode. You can passively record this, or you can then send that information to another circuit that's going to measure the deviation between the recorded voltage and whatever you set. So if you want to set your neuron to negative 70 millivolts, you can absolutely do that by just simply injecting a little bit of current to make the membrane potential more positive or more negative, whatever it needs to be to get it negative 70. And then as the neuron is receiving input at its synapses, you'll have to inject more or less current to keep the membrane potential the same. And by keeping account of how much current you inject, you'll know exactly how much current is being received by the neuron. So you don't need to know the details of patch clamp recording. You simply need to know that we can measure the current or the membrane potential, and we can also control the currents and membrane potential using just some very simple hardware. Those currents that we're measuring have to be generated by ion channels. Ions can't move through the membrane freely. This is because of that hydrophobic core in the lipid bilayer. If you've never heard of those words that I just said, you'll want to do a quick Google search and familiarize yourself with this. But because ions have charge, they're going to be repelled by the hydrophobic core, so they have to have some sort of aqueous pore that they move through, and that would be ion channels. Now there's a great variety of ion channels littered in the membrane of neurons. They all have some sort of an ion pore. This is absolutely required. Without an ion pore, ions can't move through. It's not much of a channel if it doesn't conduct ions. Ion channels are also going to have what we call a selectivity filter. This is just a collection of amino acids with certain charges and sizes that are going to allow specific ions to move through. For example, some ion channels are only going to allow sodium to move through. Others might only allow potassium, some might only allow calcium, and some might allow any ion with a positive charge. They don't care. So the selectivity of an ion channel is determined, of course, by its structure, and that region is what we call the selectivity filter. Now we get into some optional components. Some ion channels are always open. We call them leak channels. Others have an optional component called the activation gate. This is the portion of the ion that controls whether or not the pore is open or closed. So the activation gate is usually going to detect some kind of a signal like a neurotransmitter binding or a change in the membrane potential. And when it detects that signal, it pulls the ion pore open to allow ions to move. So this allows our ion channels to be turned on and off. They're not just always leaking. And this allows then the electrical activity in a neuron to be controlled in a signal-dependent manner. Some ion channels also have an inactivation gate, something that determines that the ion pore has opened and then closes it after a brief period of time. So this is sort of a little built-in self-timer, if you will, so that after ions move, that signal gets terminated, so you get very brief electrical signals. The activation gate and inactivation gate are completely separate portions of ion channels. Some have both, some will only have an activation gate. It varies from channel to channel. For example, the channels that determine the resting membrane potential have no activation or inactivation gates. They're just simply leak channels, right? Because the neuron isn't doing anything. It's not receiving a signal, so it's only leak. When we generate the action potential, we're going to thus have activation gates that depend on membrane potential, and the sodium channels have that optional inactivation gate, whereas the potassium channels don't. We'll see that that's very important in just a moment. Now, when you open up an ion channel, ions are going to move. Sometimes there's no net movement. In most cases there is, though, and there are two forces that determine the net movement of ions. For example, in this case, potassium. There's much higher concentrations of potassium inside, and so by diffusion alone, potassium would want to leave the neuron. All right, because the random motion of molecules tends to cause them to spread out. So random motion is going to push potassium out. However, recall that the inside of the cell is negative. The outside of the cell is positive. Potassium has a positive charge. Therefore, the electromotive force, so the charge interactions, are going to cause potassium to want to stay inside the cell. So there are two forces here, and depending on the membrane potential, these ions are going to move in different directions. Okay, it's really everything to do with the charge, because the actual diffusive force is constant. The actual ion concentrations really don't change that much when ions move, because these are very brief events and they're occurring only at the cell membrane, which is a very, very small fraction of the volume of the cell. So in this illustration, what we're looking at is the area of the cell that's actually receiving ion movements. I've colored it red. The cytoplasm is gray. If you look on the bottom, there's our typical cell. It has a 15 micron diameter. It has its little nucleus there, and you can see that the cytoplasm just looks gray. It's only when you zoom in on that lipid bilayer, only within about 4 nanometers or so of the lipid bilayer do you have appreciable movement of ions. And really, it's only within the first couple of nanometers. Now, 4 nanometers versus 15 microns, okay? Very, very different sense of scale here. All right, so here we're talking about a percent of a percent change. So simply opening ion channels a few times isn't going to create an appreciable change in ion concentrations. And it's certainly not going to do that because we actually have active transport to maintain that ionic imbalance. Remember, there's a lot more sodium outside, a lot more potassium inside. So whenever we open a sodium channel or a potassium channel, sodium tends to come in, potassium tends to leave. So that means that if we were to allow this to happen for long enough, we would equalize the amount of sodium inside and outside and the amount of potassium inside and outside. This doesn't occur, though, because our neuron is always spending ATP to pump sodium out of the cell and bring potassium into the cell. And that's accomplished by the sodium-potassium ATPase. This is going to use about 20% of the neuron's entire ATP. Okay, so when the neuron is generating all of its ATP in its mitochondria, 20% of that's going to go to this protein alone to help maintain the ionic imbalance needed to fire action potentials. Now, because of the very small fraction of the neuron that actually has ionic movements and the sodium-potassium ATPase, the diffusive force is constant. Okay? The only force that changes is the electromotive force. That's the only thing that's going to change on these ions. And it's going to change depending on the ion channel. Depending on the membrane potential. Now let's go through our action potential now and see how it changes and see how the movement of these different ions accounts for the membrane potential. So we're at rest. All right, let's look at our top cartoon there. We have a couple different ion channels. We have our leak channels. These are really potassium channels in this case. So they're colored red. The sodium channel in the center, that's colored blue on the inside. Its selectivity filter selects only the little blue sodium ions. The potassium channel on the right, you can see, is not a leak channel. It has an activation gate, that little green activation gate. Okay, so only whenever we're depolarized will those channels open. So at rest, potassium is leaking out. That's positive charge leaving. That creates the negative membrane potential. Let's say that we receive some excitatory input in our dendrites. That's going to depolarize the membrane potential a bit. So let's say we pull away from -70 and we hit about -50 millivolts. At that point, we have enough positive charge inside the cell to activate our voltage-gated sodium channels. These open up quickly, and when they do, sodium rushes into the cell. Let's think about what both of those forces are doing. The diffusive force, that's pushing sodium in because there's much more sodium outside the cell than inside. So by diffusion, sodium wants to come in. Sodium has a positive charge. The cell has a negative charge. Thus, the electromotive force is also pushing sodium in. So when we reach threshold potential, voltage-gated sodium channels open up and sodium rushes in. Whenever you have positive charge coming into the cell, that membrane potential is going to continue to have a positive value. So it's going to move from that -50 threshold value up to somewhere around +30, +50 millivolts. That's simply because of sodium rushing in. Now notice that that potassium channel over there also has an activation gate. Potassium channels are a little bit slower than sodium channels. So threshold potential also activates them. It just takes a little bit longer. That sodium channel is going to inactivate and that potassium channel is going to activate. So notice on that voltage-gated sodium channel in the center, it has an inactivation gate. So after a brief period of time, they turn themselves off. You're going to get that strong depolarization and the signal is terminated. Voltage-gated potassium channels will open up. Now the cell has a positive membrane potential. That's going to repel potassium. There's a lot of potassium inside. That's going to push potassium out through the diffusive force. Now our diffusive and electromotive forces are in agreement and potassium rushes out. In fact, because you have still the potassium leak and these voltage-gated potassium channels open, you have a much greater conductance of potassium and therefore you actually become more negative than you were at rest. You have what we call the hyperpolarization. Now because we're below the threshold potential, our voltage-gated potassium channels deactivate. Scratch that. They don't inactivate, they deactivate. They're very different things. The voltage-gated sodium channel in the center deactivates, and that causes it to de-inactivate. Those are both very important. So once the ion pore closes, that pushes open the inactivation gate. So that now, if we were to receive depolarization that brings us to threshold, we could fire another action potential. If the voltage-gated sodium channels don't de-inactivate, then you'll never fire another action potential. Theoretically, if you were to bring the resting membrane potential of a neuron up to, let's say, -20 or anything above threshold and hold it there, you'd get a single spike and then you'd just maintain a flat line at -20. Because your voltage-gated sodium channels never de-inactivated, we never drop below threshold. So they remain inactivated and you'll never fire another action potential. It's what we call depolarization block. It's a very bad thing. And this is one of the reasons why astrocytes are going to be there to buffer ions and neurotransmitters to prevent depolarization block. So there you have it. There's your action potential. You reach threshold, voltage-gated sodium channels open, After a little bit of a delay, the sodium channels inactivate, the potassium channels open, potassium rushes out, you hyperpolarize the neuron, the sodium and potassium channels reset, and you go back to rest, just leaking out potassium. So you get these very consistent spikes that have about the same amplitude each time because voltage-gated sodium channels open and then inactivate. So they're only briefly open. You get little spikes. And this is why we call action potentials all or none, right? Because those voltage-gated sodium channels are going to generate a very strong depolarization every time. You get about the same amplitude no matter what. What changes is the frequency of action potentials. So if you have a stronger stimulus, let's say someone is poking you lightly or tapping your shoulder very, very sternly, these feel different to us not because of the change in the amplitude of action potentials, but because of the frequency of them. So a light tap might look like the top trace there. There we're looking at little action potentials in response to a current injection, but this could just as easily be someone tapping you on the shoulder. A light tap is going to make 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17 or so action potentials. Okay, each of those vertical stripes there, each vertical tick, that's that very rapid depolarization followed by hyperpolarization. And then you can see, because we're receiving current the entire time, we continue to fire action potentials. Now, if someone pokes you very strongly on the shoulder, you get the same height for the action potential. You don't get any further depolarization because it's not possible. But because you're getting much stronger input, you tend to reach threshold much more rapidly each time, and thus you fire action potentials more rapidly. And each of those action potentials translates to the release of neurotransmitter. So whenever we have our roughly 17 action potentials versus however many that is on the bottom, there's going to be a whole lot more neurotransmitter released in the bottom. So we encode the strength of a stimulus not with the size of an action potential, but with the frequency of them. And more frequent action potentials leads to an increased release of neurotransmitter. Now let's not forget our myelin here. All right, now we know what an action potential is. It's just that positive membrane potential. How does it move down the axon? Well, without myelin there, it's going to be generated by voltage-gated sodium and potassium channels at every single step along the axon. Every bit of the axon is going to have sodium channels opening followed by potassium channels opening so that we get sodium rushing in, potassium rushing out. It's a very slow process. When we're myelinated, then we only have to perform that slow opening and closing of ion channels at the nodes. We get passive movement through the internodes. So it's just passive conduction along the axon until we reach the node. Then that depolarization that came from the previous node is enough to bring us above threshold. Once we are above threshold, our sodium channels open. We get that very strong depolarization back to max. That positive potential is going to then move passively down the node, down the internode till it reaches the next node of Ranvier, and there it's going to bring that portion of the axon above threshold potential, and so we regenerate the action potential. It's much, much faster with myelination. The whole purpose of this, though, is to communicate through little junctions called synapses. So we generate an action potential. That's wonderful, but it means nothing if we can't tell the next cell about it. Okay? So we talk between cells at synapses. There are some synapses that allow that electrical signal, that allow that action potential to pass directly to the next cell. These are what we call gap junctions or electrical synapses. So if two neurons share their cytoplasm, then the action potential just simply moves on over. This is a very small minority of synapses. The overwhelming majority are going to be chemical in nature, where we have our presynaptic site with those little chemical transmitters packaged in vesicles. When we have our action potential, we spit out the neurotransmitter, and the postsynaptic site has to wait for the chemicals to diffuse over and interact with a receptor. Okay, so we're just going to focus on chemical synapses because that's the bulk of what's going on anyway. So we have two sites that we need to think about. We're going to neglect our astrocytes and just focus on the neurons here. So we have our presynaptic site and we have our postsynaptic site. Pre means before, so this is the first part of the synapse. This is the axon, usually, where we have our neurotransmitters in their vesicles, and they're docked at what we call an active zone, and that's where we're going to release our neurotransmitter, only at the active site there. At the postsynaptic site, that contains this area called the postsynaptic density. We call it the postsynaptic density because it's on the postsynaptic site and it's very electron dense. When you look at it in an electron micrograph, if we go back a slide, you can see it's very, very, very dark on that postsynaptic site on the right illustration there. It's so dark because it's filled with proteins. Okay, so at that postsynaptic site, we have our neurotransmitter receptors. Those are proteins. Okay, those are, for the most part, just ion channels that have an activation gate that's activated not by a change in membrane potential, but by the binding of the neurotransmitter. So that when the neurotransmitter binds, the ion channel opens up to generate electrical activity. The other part of the postsynaptic density would be the scaffolding proteins that hold the neurotransmitter receptors in place and prevent them from just diffusing freely in the membrane. So we hold the neurotransmitter receptors immediately opposed to the axon there. Keep in mind, though, while we're going to think of synapses as being between axons and dendrites, there's also synapses between axons and cell bodies, axons and other axons. Okay, but this is a pretty good place to start. Now, let's go to that presynaptic site here. Let's start off with our neurotransmitters. The first thing we have to do after we synthesize them is put them into vesicles. Okay, those little spheres of protein. This is going to hold our neurotransmitters in place and allow us to release a burst at a time, and a reliable burst, because every vesicle is about the same size and they get packed with about the same concentration of neurotransmitters. So one action potential and another action potential are going to result in pretty much the equivalent amount of neurotransmitter release. Okay, so wouldn't it be nice if we just directly packaged neurotransmitters into vesicles? It would, but we don't do that. That's just simply not how it works. First, what we do is generate a proton gradient. So we spend ATP to move protons against their concentration gradient, so we fill the vesicle up with acid. And then that acid is going to want to leave because we've created this diffusive force now. When acid leaves, or when the protons leave, what they're doing is providing the necessary energy then to move neurotransmitters against their concentration gradient. It's odd that we don't just use ATP to directly move neurotransmitters, but what this does is allow us to have a single protein that spends ATP, and then we can have many other proteins that use the same chemical gradient. So we can fill a vesicle with glutamate or GABA or serotonin or dopamine. It doesn't matter. It's still the same protein spending energy. You just have different proteins taking advantage of that proton gradient. Okay, so we fill our neurotransmitter vesicles up, and then we need to get them to the right location. Okay, so once they are mature and full, they're going to exist in really two areas. There's the readily releasable pool, and then there's the reserve pool. The readily releasable pool would be those vesicles that are near the active zone and are ready to be released. They're just waiting for a little bit of calcium to come in, as you'll see. So when we fire the action potential, calcium is going to rush in, and those vesicles in the readily releasable pool will fuse. At least a couple of them will. Somewhere around 2 to 3, depending on the synapse. And there's only a small handful of vesicles in this readily releasable pool, because active zones are fairly small, so you can only fit a small number of vesicles there. So there's 6 or 7 ready to be released. Then we have our reserve pool, which can be dozens or hundreds of vesicles. They're held a few hundred nanometers away from that active zone, and once we release some of the vesicles in the readily releasable pool, then the reserve pool will send vesicles down to replenish those released vesicles. All right, so we got two locations, the readily releasable pool and the reserve pool, but both are mature synaptic vesicles filled with neurotransmitter. Okay, the whole point of firing an action potential is to cause the release of neurotransmitter, and this absolutely depends on calcium entry. So there are voltage-gated calcium channels, and those are exactly what they sound like. They have an activation gate, and it depends on membrane potential. So when that positive potential from the action potential reaches the presynaptic site, that opens the voltage gate in these calcium channels, and then calcium can enter the cell. When calcium enters, that's going to drive this process, where our vesicle here, you can see is near that calcium channel. When the action potential occurs, calcium stimulates the vesicle to fuse its membrane with the presynaptic membrane, and those little black dots of neurotransmitter are now able to diffuse out into the synapse. We know that this is absolutely dependent upon calcium for a couple of reasons. If we remove calcium or we block voltage-gated calcium channels, we don't get those excitatory postsynaptic currents. What I want you guys to look at here is just that top trace. EPSC means excitatory postsynaptic current. Excitatory means that it's going to cause the neuron to be excited. It's going to depolarize it closer to threshold. So what you'll notice is that there's that downward deflection. That's showing you the movement of sodium into the cell. And this occurs unless we apply cadmium. Cadmium will block voltage-gated calcium channels. So you'll notice that that little dotted line doesn't have the same downward deflection. And that's because without the activity of voltage-gated calcium channels, we can't generate these postsynaptic currents because we can't release neurotransmitter. Conversely, if we simply release calcium using light rather than the action potential, we can actually stimulate neurotransmitter release without using action potentials. So they used this special form of calcium that's trapped in a little chemical cage, and you can shine a light on that calcium to break open the cage. Okay, so there's a couple panels here. Delta F over F is just telling you the change in fluorescence. So the change in light, if you will. As there's more fluorescence, so there's the thin solid line, there's the thick solid line, and there's the dotted solid line. By shining with different powers of laser, you can generate brighter light, and that's going to translate to more intracellular calcium. Let's look at the second trace. Again, straight line, very brief increase in calcium. The thick line, a little more prolonged increase in calcium, and the dotted line, which had the brightest light, has the most calcium. Now let's go to the bottom. Here's where we're looking at our EPSC, that excitatory postsynaptic current. The solid line, the thin one, you can see a very small inward current. That translates to the lowest level of calcium. The thick line, the intermediate level of calcium, gives you an intermediate excitatory postsynaptic current. And the dotted line, where we had the greatest amount of calcium increase in the cell, gives you the greatest postsynaptic current because you had the greatest amount of neurotransmitter release. Now the release of neurotransmitter is regulated by calcium, but it's carried out by a set of proteins that we call the snare complex. These are the same snare proteins involved in moving vesicles around within the cell, like between the rough ER and the Golgi. So you can dust off those cell biology books that you have and refresh yourself on snares here. All you need to know for this class is that snare proteins are attached to the vesicles and the presynaptic membrane, and when calcium enters, those snare proteins zipper up with one another, releasing a sufficient amount of free energy to drive vesicle fusion. Okay, so when calcium enters, snare proteins zip up, vesicles fuse. Just that simple. We care about three proteins here. There's the snare protein on the vesicles, that would be synaptobrevin. It's also called VAMP. This is going to be embedded in the vesicle membrane. You can see the little purple vesicle that's docked in the upper left corner of the illustration, and you have your blue synaptobrevin attached to the vesicle. You have your purple syntaxin that's going to be attached to the presynaptic membrane, that's green, and that's also going to contribute one single helix into the snare complex. And then these two helices, one from synaptobrevin, one from syntax, are held together by two additional helices from SNAP25. So SNAP25 is the last snare protein. That's going to essentially snap together the snare complex and hold it. It's not attached to either the vesicular or presynaptic membrane, but instead it's just there to hold the snare complex together. When calcium enters, then we proceed to the upper right and then to either one of the lower portions. There may very well be a transient protein-lined fusion pore where the snare proteins make a pore. That's debatable. Without a doubt, there is fusion of the membranes, and you do get that lipid-lined fusion pore. And eventually your vesicle becomes continuous with the presynaptic membrane. You fuse the membranes. That means that we're going to run out of vesicles if we don't recycle them. Okay, so our presynaptic site has a very highly functioning recycling system in place, and you need that because your typical presynaptic terminal is going to have a couple hundred vesicles, and that might sound like a lot, but it's really not. Every action potential is going to burn 1 or 2 vesicles. Okay, because the 6 or 7 that are docked, not all of them are going to fuse. You'll get 1 or 2. So that means if our neuron is firing at 10 hertz, which is totally not unheard of, 10 times per second is well within the range of possibilities. Some neurons fire at 100 hertz, so 100 times per second. Crazy fast. But even at a mere 10 hertz, you're going to run out of vesicles in about 20 seconds. So that means you have about 20 seconds to live until it's all over, unless you recycle those vesicles. And there's a few ways that recycling occurs. We're not going to go over any of the details here. You just simply need to know that they exist. One very fast method of recycling is what we call kiss and run. So you form that fusion pore, but you don't actually completely fuse your vesicle with the presynaptic membrane. You form just the transient protein line pore, and then you snap off. But before expelling all of your neurotransmitter, then you refill to top up. That's kiss and run. So you just have a brief kiss with the presynaptic membrane to spit out a little bit of neurotransmitter, and then you're back. Merge and recycle is kind of the classical view of neurotransmitter release. The vesicles fuse completely with the cell membrane, and then after a little bit of cleanup, we're going to pull back a vesicle and refill it. If you have a large number of vesicles fusing, you might need bulk endocytosis. That's the last mechanism there. It's on the far right of the illustration, where you're going to pull in a large amount of protein that you'll then break off into individual vesicles within the presynaptic terminal. No matter which way we go about it, we have to recycle our vesicles because we have a very finite number of vesicles available, and we burn through them because neurons are active all the time. All right, an inactive neuron, or one that's inactive for a prolonged period of time, is a dead neuron. So we generate action potentials so that we can release neurotransmitter, and we release neurotransmitter so that we can generate currents in our target cell. That target cell might be a muscle. We might be asking our muscles to contract so we can run away or pick up a sandwich and eat it or whatever it may be. Or it may be another neuron. Okay. So in our neurons, we can either have excitatory or inhibitory postsynaptic currents or potentials. They're pretty much the same thing. An excitatory postsynaptic potential is going to depolarize the membrane potential. So if we look on our cartoon setup here, what we're doing is recording the membrane potential in the cell body after we receive these inputs in our dendrites. There's 2 excitatory inputs. They're both colored yellow, and there's 1 inhibitory red input. You can see that the yellow synapses are going to cause the membrane potential to become more positive in panel B there. So in that first section, we're getting an excitatory postsynaptic potential. Let's say only one of our synapses is active, one of our yellows, we get a little bit of excitation. And really in the central nervous system, all the synapses are incredibly weak. So you need many synapses to be active at once in order to actually depolarize to threshold. But in this cartoon example, we just need those two synapses to fire together. When that happens, the excitation sums, and we cross threshold and we fire an action potential. Great. That's the purpose of excitatory postsynaptic potentials, to stimulate the firing of an action potential. But you usually need many of them to occur together. The red synapse, that's going to be our inhibitory synapse, that hyperpolarizes, or in most cases it actually just prevents depolarization through what we call shunting inhibition. But if you look on the middle part of panel B, you can see when our red synapse is active, we get a little hyperpolarization. Really, the inhibitory currents are going to have not so much a hyperpolarizing effect, but what they'll do is resist changes in membrane potential. So if you look over in the next two portions, the orange is showing you the sum of our excitatory and inhibitory currents. So we take that little bit of hyperpolarization, that little bit of depolarization. What you notice when you put them together, what happens is we just get less depolarization. So even when both of our excitatory synapses are active, that should bring us above threshold. That's what the yellow is showing us. We should go above threshold, but we don't. If we have just that little tiny inhibitory input, that's going to resist changes in membrane potential and typically prevent the firing of action potentials, as it did in that little orange trace. Now, not every neurotransmitter receptor is going to create currents in the same time span. Some do it very quickly. Those would be the ionotropic receptors. They're just ion channels. That's it. Some are going to be sodium channels, some will be potassium channels or chloride channels, some will be cation channels. Some will allow calcium to come through, which calcium is an important secondary messenger that can control the activity of enzymes. But the ionotropic receptors are really just ion channels with an activation gate dependent upon a ligand. Metabotropic receptors are very different. They're not ion channels. They are tethered to usually G proteins, which are these little intracellular proteins that control their target enzymes. And what you instead get is this signaling cascade. So when you see something like this, you should be thinking, this is going to take a while. This is much slower than just opening an ion channel. So here we don't generate fast electrical currents. Instead, what we're going to do is cause slow but long-lived changes in cellular signaling. So we activate our receptor. That one receptor can activate many G proteins. Each G protein is going to then act on its target enzyme. In this case, they're using adenylate cyclase. That's an enzyme that's going to create cyclic AMP from ATP. So we have another round of amplification where each adenylyl cyclase is generating many molecules of cyclic AMP. Each cyclic AMP molecule will bind to protein kinases. For example, protein kinase A is activated by cyclic AMP, and then active protein kinase A can then go phosphorylate many targets. So you have another round of amplification. So a single receptor can lead to a change in the activity of many proteins because of this signal amplification. Some of these targets are going to end up being ion channels, so we'll still generate an electrical signal. It'll just take a while to come along, and it will usually last a little bit longer. Sometimes the targets are going to be transcription factors, and so these metabotropic receptors actually lead to a change in gene expression. Whenever we're thinking about fast synaptic transmission, we're just thinking ion channels. That's it. All right, keep it here. The neurotransmitter binds, we get a very brief electrical current, that neurotransmitter gets cleared, and we're good to go. Keep in mind, though, that some neurotransmitters can cause very long-lived changes in cellular function by activating metabotropic receptors. Now remember, after we release transmitters, we can't let them hang around all day. We got to clean them up. Okay, so there's those astrocytic transporters, and if you remove their glutamate transporters, you get lethal seizures. Neurons also have transport proteins, okay, so that after release, they're going to be removed from the synapse, either by neurons or glia. Not every neurotransmitter has transporter proteins, though. For example, acetylcholine doesn't have a transporter. There are choline transporters, but the way that we clean up acetylcholine is by degrading it enzymatically. So there's this enzyme, acetylcholinesterase, that's going to break down acetylcholine. This way, when we release acetylcholine, our muscles can contract, and then that enzyme can degrade acetylcholine. Not every neurotransmitter has an enzyme that degrades it, though. We don't have enzymatic degradation of glutamate or GABA, the two principal neurotransmitters in our central nervous system. So some neurotransmitters will have a mix. For example, serotonin has transporter proteins and it has enzymatic degradation. Others only have transporters, as is the case with glutamate and GABA. Others only have enzymatic degradation, as is the case for acetylcholine. So we can regulate the level of neurotransmitters after they're released. Now, of course, we can also regulate how much neurotransmitter we are releasing. Now, while we're going to think of synapses as typically between axons and dendrites, that's not always the case. Sometimes axons will synapse onto other axons to control the degree to which they're going to release their neurotransmitter. So some axo-axonic synapses, as they're called, are going to cause presynaptic facilitation, where they're going to actually increase the release probability by promoting calcium entry. So what they could do is activate ionotropic receptors that depolarize the presynaptic site to facilitate calcium entry, or they could activate metabotropic receptors that sensitize voltage-gated calcium channels or modify snare proteins to facilitate release. And of course, there's also presynaptic inhibition that decreases release probability. You could activate some of those inhibitory receptors that's going to resist changes in membrane potential and therefore reduce the amount of calcium that enters, or you could modify the function of voltage-gated calcium channels through metabotropic receptors. So both of these options are possible, where we can increase or decrease the function of a synapse simply through these axo-axonic connections. And then, of course, there are receptors that are going to be activated at the presynaptic site following neurotransmitter release. Okay, now this is not the same thing as an axo-axonic synapse. Here's where we have the neurotransmitter that's released at that synapse acting at receptors at the presynaptic site. So those would be autoreceptors. Auto just means self. So these are receptors at the synapse that recognizes its own neurotransmitter. These tend to act as inhibitory receptors that then either prevent the synthesis of the neurotransmitter or its release through some mechanism. The logic here is that if you have very high levels of neurotransmitter release, that's going to cause the activation of autoreceptors and thus tone down the amount of neurotransmitter release so that you don't hyperexcite your target cell. So in this case, we're looking at two presynaptic terminals, and they're both releasing neurotransmitter. If that neurotransmitter binds to a receptor on the same presynaptic site, that's called an autoreceptor. If it diffuses over and binds to a presynaptic receptor on a different presynaptic site, then it's a heteroreceptor, which just means other. So both autoreceptors and heteroreceptors, they could be the same protein. It just depends on the source of the ligand. And both of them are going to tend to be inhibitory in nature, because they'll only be activated if you have sufficiently high levels of neurotransmitter to escape enzymatic degradation or the reuptake mechanisms that are built in to control neurotransmitter levels. So if you have hyperexcitability in your presynaptic neuron and it's releasing way too much neurotransmitter, those autoreceptors will help tone that down to prevent the postsynaptic cell from becoming damaged and hyperexcited. Okay, ton of stuff there. Email me if you have any questions, but make sure you're familiar with the anatomy of neurons, the function of all the different glial cells, the resting membrane potential, and the role of leak channels in creating it, that you understand voltage-gated sodium and potassium channels and their role in the action potential. And then make sure you understand how calcium drives neurotransmitter release and how neurotransmitters then generate fast postsynaptic currents through ionotropic receptors, or that they can generate slower, more long-lived changes through metabotropic receptors. Okay, email me your questions, and I look forward to seeing you for Chapter 3.